So I did my project on ATP synthase. So oxidative phosphorylation is composed of 5 complexes, and its goal is to produce ATP. And in complexes 1 through 4, electrons from NADH and FADH are being transferred through the 4 complexes with the help of mobile electron carriers, coenzyme Q, and cytochrome C. At the same time, protons are being pumped out of the matrix into the inner membrane space through complexes 1, 3, 4, which act as proton pumps. This creates a change in the pH within the matrix, and the electric and chemical gradients and changes fuel the process of ATP synthase in complex 5, where ADP and phosphate are converted into ATP. So ATP synthase is the enzyme that converts ADP and phosphate into ATP. And it's located in the mitochondria of cells of eukaryotes and plasma membrane of bacteria. It's the final enzyme in the OP pathway, and it's composed of 20 subunits that are a mix of soluble and integral proteins, and it's divided into 2 parts, F0 and F1. And its molecular weight is about 550 kilodaltons. The F0 portion is a proton turbine responsible for the rotation of the central stalk in F1 and drives the phosphorylation of ADP. It's bound in the inner mitochondrial membrane and is involved in proton translocation by acting as a proton channel, as the protons that are in the IMFs come back into the matrix. F0 is powered by protons flowing back into the matrix and the mitochondria, while the F1 is catalyzing synthesis of ATP. The F1 portion has 3 catalytic sites, is a molecular motor that drives rotation of stalk with ATP hydrolysis, disruption of the interactions of the central stalk is predicted to cause altered efficiency of the enzyme or uncouple the enzyme. And it's found in the mitochondrial matrix and is water soluble. And the image right there is just describing like the 3 catalytic sites and conformations that it can contain in the beta subunits. So in the first part, substrate is bound to the open site and is converted to ATP at the tight site. In step 1, the rotation of the gamma causes a conformational change, and this results in the change in formation of the sites. So ATP is released from the enzyme. And then in step 2, substrate again binds to the open site and another ATP is synthesized at the tight site, and this just keeps going on and on. So in my literature, I found a paper that was trying to identify if antibacterial properties of ginger phenolics are linked to the inhibition of bacterial ATP synthase. And just phenolics are just different structures, like different types of ginger, which are listed at the right. And it's known for its anti-cancer and anti-inflammatory properties, and a variety of drugs or other phenolics are proven to bind and inhibit F0, F1 ATP synthase. So they want to see if these ones would do the same. So they took mutant and wild and null E. coli strains and incubated them with F1, F0 ATP synthase. And these E. coli strains either contained the ATPase gene or they didn't. And after they took the wild and mutant type ATP synthase and incubated that with the ginger phenolics for 1 hour at 37 degrees Celsius in a buffer. And the optical densities were from this that were measured were compared to the phenolics. It was compared to the samples without the phenolics at the optical density 700, and they determined the optical density of 700 of the samples with the phenolics using this. And they overall found that all the wild type membrane bound F1, F0 ATP synthase had a 90 to 100% inhibition for the bacteria. Sorry, tongue tied. And then this is just like a chart that they included with the different like strains, mutant strains that they added to the different phenolics. And they included the concentration of the DGPs in micromolar. And this is my slide or my references.